test cross. So here a new concept, test cross. The word test, this is what we are going to do. We are going to test the genotype. Now think we have an organism, a plant. We can see it. We can see the morphological features. We can say it has purple flowers, it is tall or whatever. The morphology we can see, so we can tell. But the genotype, we can't. Sometimes we can say the genotype if we know some details about the plants. Sometimes we can't say the genotype, right? So now think a situation. For as the example, I will take we will take the height, right? The height of a plant, right? Now we know we normally take this capital T and simple T for the height. So we have the capital T dominant allele and simple T recessive allele. If the genotype is simple T and simple T, then we know that it is a dwarf plant. Then if the genotype is capital T and capital T, or capital T and simple T, then the phenotype is tall. So this is the basis. Now think we have a plant and we know we should we need to predict the genotype. The plant that was given to us is the plant that was given to us is, if it is dwarf, if it is short or dwarf, then we can say, we can directly predict and say that the genotype must be simple T, because we know dwarf plants are, or we get dwarf plants only if the genotype is homozygous recessive. So if the plant is dwarf, we can say the genotype of that plant is definitely simple T. But if the plant that was given to us is tall, then can you say exactly what is the genotype? We can say it can be capital T and capital T, but there is a chance that it can be capital T and simple T as well, right? Or we can say that it must have at least one dominant allele, one capital T allele. But we are not sure about this, the second allele. So we can't definitely predict and say what is the genotype of that tall plant. So this is a question. What is the genotype of this tall plant? So it is a question. We need to find that using a cross. That cross is known as the test cross, because we are going to test the genotype of that plant with that unknown genotype. We are going to find that, right? So that sort of a cross is called a test cross. So now we will see how to do a test cross. Right. So Right. So we have two options. We have the genotype can be either capital T, or capital T, simple T. Right. Now we are going to find it by crossing that plant with a double recessive plant for that condition. We are going to do a cross. We are going to cross this unknown genotype and the genotype plant with a double recessive with a plant with a double recessive condition. That means with a dwarf plant. Dwarf one, we can, by looking at the phenotype, we can take the plant. We know that it is simple T. Right? Now we are going to show that using the letters. Now we have two options, two uh, ways, capital T, and the other one is capital T, simple T. So first one, here the genotype is unknown, right? Now we will see how to find that. We are doing a real cross now. Thing, we have taken this first one, capital T, double dominant plant. Think this first one unknown one has capital T. Then now we will see what are the gametes we get and what should be the F1 generation. Here the gametes, this one is the parent. You have to write that T1 generation. Then the gametes are here, it should be capital T. Then in the other side, the gamete should be simple T. Then when they fuse together in the cross, we get capital T, simple T, 100%. All of them are capital T, simple T. And because they have capital T, all of them should be tall plants. So in the F1 generation, we should get 100% capital T, simple T, tall plants in that situation. Now, then we will go to the second condition. That is, if the genotype of our unknown is capital T and simple T. Now, this one is the unknown genotype one that we are testing. We cross that with, again, we are doing the same thing. We cross that with a dwarf plant, double recessive genotype. So this is the parental generation. So we get, as the gametes, we get two types of gametes, capital T and simple T gametes. Here in the other plant, we get only one type of gamete, simple T gametes. Then in the F1 generation, this capital T goes with simple T, capital T, simple T. And then this simple T goes again with simple T. So it is simple T. So we get two types of two genotypes, right? Capital T, simple T, that should have the phenotype tall, and the other one, simple T, should have the phenotype dwarf. So what has happened in this situation? We get 1-to-1 ratio of, or else we can say 50% of the plants are tall and 50% of the plants are dwarf. That is in the F1 generation, right? So this is how we find the unknown genotype. Now think we did the cross, and in the F1 generation, if we get all the plants 100% percent tall plants, then we can say that our unknown genotype, the plant is capital T, capital T. Then the second situation in the F1 generation, if we get 50, half of the plants tall and half of the plants dwarf, then we can say our unknown genotype is capital T, simple T. So like this, in this way, we can predict the genotype of an unknown. That sort of a cross is called a test cross. And here we used only one character, right? Like in that Mendel's experiments, when we use one character, we use only the height of the plant. Then it is known as a monohybrid, right? In monohybrid cross, but here it is specially a test cross. So this is called a monohybrid test cross.